today's my topic of discussion is the next generation sequencing and application in the forensic science. As you all know that this is the my contents of my today's presentation, role of DNA analysis in forensic science, development of DNA analysis, advancement in technology, next generation sequencing, and the future of DNA analysis. So first we are discussing about the role of DNA analysis in forensic science. So mainly, what are the role of DNA analysis in the forensic science? The first one is to identifying the perpetrators. Okay, for example, there are some kinds of crime has been occurred. So if there are any kinds of body fluids are present in the crime scenes, so either is the saliva, semen, or the blood. So definitely, you can extract the DNA from the body fluids and match with the suspects. Okay, so this is the matching the DNA profile. Matching the DNA profile is between the, it helps to link the crimes. Matching the DNA profiles is helps to linking the crimes as well as exonerating the innocent. So what about the exonerating the innocent? So in case of exonerating the innocent, someone claimed that these individuals are involved in this crime, but on the basis of the DNA evidence, we can exonerating the evidence because there are any traces of DNA are, cannot be found in the victim body. In case of rape cases, so what happens in case of rape cases? So definitely there are some kinds of traces of semen are found, seminal fluids are found in the victim body. So that if we are going to extract the DNA from the semen, definitely it's some kinds of link between the victim and with the perpetrator. And this one is the linking the crime. So what about the linking the crimes? So in linking the crimes is the identifying the serial offenders. So what about the identifying the serial offenders? Means there are, from one crime scenes we are getting one types of body fluids. There is another crime scenes we are getting another types of body fluids, and from those of the body fluids we are extracting the DNA, and from the DNA analysis we are inferred that both of the DNA are belongs to same individuals. So definitely, so those individuals, these are the particular individuals who committed the crime repetitively. So that's why it's identifying the serial offenders. So this one is the second, and the third one is the identifying the victims. So in case of mass disaster cases and the missing persons, so if we have some kinds of DNA database of the individuals, so definitely in mass disaster cases, so what happened, there are so much people are missing. So to identify the identity of those missing persons, so what we can do, so just for example, we are extracting the DNA from any body fluids of those individuals, or we can extract the DNA from the bones of those individuals, teeth of those individuals, and then what happened after extracting the DNA, we are matching with their close relatives. It's either match or not. Okay, so this one is the identifying the victims. Next, importance of DNA in the forensic science. So establish the paternity and the kinship. So the DNA analysis helps to determine the paternity. So what about the determine the paternity? In case of determination of the paternity, there are some kinds of paternity disputed cases. So someone have, where in paternity disputed cases where the child's particular child's fathers or the child's paternities are in question, that this the A individuals are the child parents, or the A individuals is the child father or not. So to determine the paternity of those child, so we are going for the DNA analysis and any other familial relationship which can be crucial in legal cases. Next one is the historical and the archaeological investigation. So where we get the ancient DNA. So in ancient DNA analysis of ancient remains provides insights into human history, migration pattern, and the genetic disease. So what about the development of the DNA analysis? So the DNA analysis, to extract the DNA, definitely there is a weird requirement of the cells. Without the cells, we cannot extract the DNA. It's either blood cells or buccal cells. Any kinds of cells is required for extract the DNA, because the DNA are present in the nucleus, within the nucleus of the cell. So DNA analysis or starts from the 1953, where James Watson and the Francis Crick discovered the double helix structure of DNA. So as you all know, what about the double helix structure of the DNA, and the laying the foundation for understanding the genetic informations. So after the after the discovery of the structure of DNA, what happened? So the DNA are revolutionized, DNA analysis are revolutionized in 1984, where the Alex Jeffries development the technique of DNA fingerprinting. So what about the fingerprinting? So fingerprinting, in our hand there are some kinds of regions are there. So definitely this is the fingerprint. So the fingerprints is the unique identity of the individuals. So it cannot matched. Okay. So definitely the Alex Jeffries developed the techniques of DNA fingerprinting. So on the basis of the DNA, we can identify the individuals, because the sequence of the DNA are unique to individual. So which involves analyzing the variable number of tandem repeats in DNA. So what about the variable number of tandem repeats? So in variable number of the tandem repeats, there are some kinds of repetitive sequence of the DNA. Okay, variable number, there is some kinds of repetitive sequence of DNA. This is the variable number of tandem repeats. And there, for example, there is a 10 repetitive sequence of the DNA, 12 repetitive sequence of the DNA. Okay, so this is the variable number of tandem repeats in DNA. This technique was first used in forensic investigations in 1984. So this technique, so VNTR technique was first used to in forensic investigation, means on the basis of this variable number of tandem repeats, means someone have the 5 repeats, someone have the 10 repeats of this particular loci. So on the basis of this repeat sequences, we can differentiate the individuals. So what are the revolutionized in the forensic science? The first use of DNA evidence in criminal case occurred in the UK, which leading to the convictions at the 1986, and the 1987 the first DNA evidence was admitted in a US court, further solidifying its importance in legal proceedings. So what about the technological evidence in the DNA sequences? The first, the earlier one, before the invention of the PCR. So what we do? So extract the DNA, and then so entire length of the DNA is contains 3 billion base pairs. So in earlier one, we are going for the RFLP. Just cut the DNA into several fragments. So there is a using of some kinds of restriction in them, which cut the DNA into multiple fragments. So this is the previous techniques. So after the invention of the PCR, so the PCR, what the PCR did? So the PCR is just like a copier. Okay, just like a copier. It's like a copy machines. So the PCR, what happened in the PCR? PCR amplified the small DNA samples. What happened? The PCR amplified the small DNA samples. There is a 3 billion base pairs of the DNA. For example, we need only the particular 10 base pair region of the DNA. Okay. So what it did? There is a requirement of the primers. So the primers attached with this sequence and the cut the particular region of the DNA, and then they amplify it. So this is the PCR. Then there is a short tandem repeat analysis are there. So in short tandem repeat analysis, STR analysis become the gold standard for DNA profiling. So because in the STR analysis is a short tandem repeat, okay. So what about the repeat tandem repeat? So there is the same kinds of sequence appears repeatedly. This is the tandem repeat, same kinds of sequence, for example ATGC, 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 the same sequence appear repeatedly. So this is the tandem repeat. And what about the VNT and variable number of tandem repeats? So the variable number of tandem repeats is mainly the thing is that in the variable number of tandem repeats where there is a repeat length on the basis of the repeat length, okay, and the number of bases. So number of bases is much more, base pair is much more in case of VNTR is more than the 6 base pair. In the VNTR in generally is more than the 6 base pair, okay. But in case of STR short tandem repeat, okay, so there is some kinds of short sequence which appear repetitively. So in short STR, so the length of the sequence is only the 2 to 6 base pair. So length of the, so that's it is the difference between the VNTR and the STR. So in case of VNTR, the length of the sequence is more than 6 base pair, but in case of STR, the length of the sequence is only the 2 to 6 base pair. So these kinds of repeats. So on the basis of this STR analysis, so what we get in the STR analysis, for example, there is a 3 individuals are there in the particular loci. So some individuals have 11 STR repeats, some individuals have 12 STR repeats. So on the basis of these differences, we can identify that both are not the same individuals, both are the different individuals. So that's why STR is the gold standard for DNA profiling due to its high sensitivity and the discriminatory power. So because it's a very much short, number of base pair is less, so that's why the STRs have much more discriminatory power. So then we go for the next generation sequencing. So the NGS revolutionized the DNA analysis, enabling the rapid, comprehensive and cost-effective sequence of entire genome. So what about the NGS? So we are discussing in the later. So this is the steps of analysis of DNA. So what are the steps of analysis of the DNA? The first thing is that forensic laboratory receive crime scene evidence, then they are going for the perform the preliminary test. Okay, so if you are getting the blood or if you are getting the saliva or the semen, so what about the preliminary test? We are going for the some presumptive test to identify that this one is the blood or this one is the saliva, this one is the semen. So if they,